Morning, everyone. So for those who show up in the first talk on the third day, you must be truly expert in Finnish calculations. I don't need to convince you about the accuracy, like the modern implications of Finnish calculation can achieve, and how the accurate calculation can possibly impact small molecule drug discovery projects. But I do want to start by reminding everyone working on methods development how important it is to test your method on a large number of systems. We can all fool ourselves by looking at a small number of data points. So to facilitate data curation and methods comparison, we have collected by far the largest publicly available data set for protein small molecule binding potency calculation. So the data set covers more than 60 chemical series binding to broad class of protein targets, including R group perturbation, scaffold hopping, charge perturbation, microsatellation, and so on. So I encourage everyone working methods development to test your method on the entire data set to get a realistic assessment of accuracy. So accurate protein ligand binding potency prediction is great, but as you all know, drug discovery is much broader than small molecule potency prediction. So how can Finnish calculations help other problems in drug discovery? For example, for many targets, particularly GPCRs, ligands with similar binding potency can have very different function response. Some can activate or enhance the receptor function, while others can inhibit or block receptor function. Those are called agonist and antagonist, respectively. So how do we model the function response upon ligand binding? Another example, over the past few decades, the market share for peptide and antibodies has been increasing rapidly. So how can we use Finnish calculations to help discover peptide and antibody drugs faster and cheaper? And more importantly, how do we obtain the pH-dependent binding? Another example, so after obtaining the development candidate, how do we choose the most appropriate formulation for the final drug product, the crystal structure prediction, solubility calculations? So over the past few years, we made rapid progress in all these areas. So let's dive into each topic in detail. First, about the function response modeling. As mentioned earlier, many targets like GPCRs, potent molecule may not have the desired function response. Some act as agonist, others act as antagonist. So nearly for such targets, potency optimization is not good enough because you may not have the desired function response. So on the molecular level, the mechanism for the small molecule to act as agonist or antagonist is following. So in the apo state, there is a basal distribution between the ensemble of active versus inactive state. So in the presence of ligand, in the hollow state, the distribution between the active versus inactive state will shift. Agonist will shift distribution towards the active ensemble, while antagonist will shift distribution towards the inactive ensemble. So the time scale for the conformation change between different protein conformations is too long for practical modeling. The question is, how do we model, how do we predict the function response upon ligand binding? To address this problem, we came up with the following thermodynamic cycle that collects the different conformations of protein relevant for its function with ligand binding. In particular, in the apo state, the distribution between the active versus inactive state is determined by the energy difference between those two conformations, that is the apo. A small molecule may interact with the protein in the active or inactive state, but interaction energies usually differ. Because of different interaction energy with the ligand, the distribution between the active versus inactive state will shift in the presence of ligand. So if the function response of receptors is determined by the thermodynamics, then it's very easy to prove that the relative shift between those two distributions in the hollow state versus apo state is fully determined by the relative interaction energies between the ligand with the two protein conformations. That is, delta G hollow minus delta G apo is equal to delta G A versus delta G I. Well, delta G A and I refers to the interaction energies between the ligand with the active and the inactive state of protein, respectively. So based on this model, For each ligand, you just need to run 2 absolute binding FEP calculations to calculate its interaction affinity with the 2 receptor conformations. And then the ABFE score difference will determine the functional response upon ligand binding. Let's take a very simple example. See, for the beta-2 receptor shown here. So here we have 2 structures, each with the agonist and antagonist bind respectively. Then for an unknown ligand, there are multiple for which we want to predict the functional response upon ligand binding. We just need to dock this ligand in its 2 receptor conformations and run 2 ABFP to calculate the interaction affinity. And according to our calculation, there are multiple interacts with the active state of protein more favorably by about 6.7 kcal. So our model predicts this molecule is a strong agonist. And indeed, experimentally, it is the beta-2 agonist to treat asthma. And we have collected a large number of systems with experimentally determined functional response on a diverse set of ligands. And then we run the same ABFP to calculate the interaction affinity between each ligand with the 2 receptor conformations. And very surprisingly, the ABFP score difference can perfectly explain the functional response upon ligand binding. So in this functional response seesaw plot, the y-axis is the ABFP score difference between each ligand with the 2 receptor conformations. Each bar represents one ligand, color-coded by the experimentally determined functional response. Orange bars represents agonist, blue bars represents antagonist from experiment. Take the beta-1 receptor, for example. All the ligands predicted to have stronger interaction affinity with the active state of protein corresponds to experimentally determined agonist, while the reverse is true as well. With 100% classification accuracy, it's almost true for all the targets we tested so far, including 1 nuclein receptor. The results indicates that the receptor function response is indeed determined by thermodynamics, and the ABFEP between the 2 receptor conformations can accurately predict the function response upon ligand binding. And I want to highlight that our model also works very well for very tricky SCR, well, very small chemical modifications in the ligand affects the function response between different acid forms of same receptor family. For example, for those 3 ligands with very small R group modifications. The first one is antagonist for both delta and mu opioid receptor. Second one becomes agonist for delta, while the third one becomes agonist for both receptors. And those effects are all accurately modeled. Another example for this ligand LJ17, it has hallmarks of both adenosine agonist and antagonist, but the function response is also accurately modeled by our method. For those very different scaffold binding to serotonin receptor, some act as agonist, others act as antagonist. Again, our model accurately predict the function response for all those scaffolds. So all those cases are extremely challenging for machine learning or cheminformatics types of methods, but our physics-based modeling can accurately predict the very subtle SCR. And all the results I just presented were reported in this paper, which you can read for more details. And I just want to mention that right after our publication, a research group in Japan also applied the same method to predict the conformational selectivity of kinase inhibitors and also obtain the perfect classification accuracy, similar as ours. Just a short summary for this section. So we have different methods that can accurately, reliably predict the function response upon ligand binding. The minimum information required is the structure of the active and inactive state of protein, and then the ABFEP calculations between the 2 receptor conformations can predict the function response, and all the script to set up and analyze results are available to download from the paper. Now let's switch to the second topic about peptide. As mentioned earlier, over the past few decades, the market share for peptide drugs is increased rapidly, and the blockbuster GLP-1 peptide drug is only making the field much hotter. So very interestingly, the majority of the approved peptide drugs are selective peptide. Maybe microcirculation is required to restrict the conformation space of peptide to obtain the potency and permeability. And microcirculation also poses a significant challenge for sampling the conformation space of peptide. So very often, like backbone modifications like introducing additional cycles or bridges can change the binding mode for a bioactive conformation protein significantly. And those are too challenging for relative binding affinity calculation. So in the following, I'm going to focus the calculation on the side chain modifications for cyclic peptide. So here's one example. It's the breakthrough PCSK line cyclic peptide inhibitor from Merck. Experimental SCR available for a large number of side chain modifications from this paper. This figure shows the F+ performance from the default protocol on this system. As mentioned earlier, the bioactive conformation of peptide in binding pocket usually differs from that in the lowest energy conformation solvent. To improve the conformational space sampling, we included the entire ligand in the rest region in the solvent leg of FPP and also run transformation longer to about 15 nanoseconds. And with this kind of enhanced sampling, the experimental SCR is accurately modeled by the method. Similar story for the cyclic peptide inhibitor, blocking the NRF2 KEAP1 protein-protein binding interface, again with enhanced sampling of ligand. And through rest and longer simulation time, you can see that experimental SCR is perfectly explained by the model. Here's the kind of more complex example. It's the cyclic peptide mimicking the beta hairpin, blocking the P53 HDM2 protein-protein binding interface. Experimental SCR we reported for a large number of side chain modifications in this paper. The majority of many of the variants involve multiple side chain modifications, and the chemical modification is too big for a single step FPP. So here what we did is very simple. We just separate the ligands into different subseries according to their chemical similarity. So in all the perturbations, we only include one side chain modification in each edge. You can see that F+ performance for most of the subseries is looking pretty good. For a few, like the yellow one here, is kind of a bigger error in FPP. But those kind of accuracy is good enough in our experience to drive the prospective design work. We also have encountered much more complex examples like the switch-to domain of PRAS. This pocket is known to be very flexible with very small R group extensions on the peptide. It pushes alpha helix away from the binding pocket and opens a different sub-pocket. So here, since we know the existence of those two protein conformations, again, we class the ligand into different subseries according to their similarity with the two co-crystals ligand. F+ performance for the first subseries is pretty good, but the second one is suboptimal. And this kind of system is most challenging because it's very likely that some of the side chain modifications can also push the pocket to arrange in a different conformation that has not been observed in experiment yet. So we are in the process of combining FDMD with protein reorg FEP to predict such conformational changes in the binding pocket and to calculate the phenotype associated with the conformation change of protein. A short summary for this part. For cyclic peptide, most often the side chain modifications won't affect or won't change the binding mode or bioactive conformation of peptide significantly. And with enhanced sampling of ligands through rest and longer simulation, those modifications can be accurately modeled by F+. For some flexible pockets, even the side chain modifications can induce binding pocket conformation changes. And for those kind of cases, most often, if we carefully separate ligand into different subgroups according to their most likely preferred protein conformation, reasonable accuracy can also be expected. But for very challenging cases, like either backbone modifications or binding pocket conformation changes induced by side chain modifications, that is unexpected. So those requires further development of enhanced sampling to make it more reliable. Similar story for antibody drugs. So the market share for antibody drugs has been increasing rapidly over the past many years. To assess the accuracy of protein FPP for protein-protein binding potency prediction, again, we collected a large number of systems with reliable experimental binding affinity measurement for more than 250 cytosine mutations. The dataset covers small peptide, antibody, and other protein-protein binding interface. So here's the results. So the RMC between prediction versus experiment is 1.35, not perfect accuracy as compared to small molecule binding, but still you have a pretty high classification accuracy, and only less than 10% of the mutations has error bigger than 2. And more importantly, after carefully analyzing the outlier cases, we have developed an empirical method that can flag what kind of mutations are likely outliers in FTP calculations prior to the calculation. Say, for example, on this very large data set, after excluding the cases where we empirical method identifies likely outliers and also introduce a one-parameter correction for the charge breaking mutations, the RMC for this large data set reduced from 1.35 to 1.02, and with perfect classification accuracy. And we expect such kind of a semi-empirical method to be very important in prospective study for protein FTP because it will help users to guide the decisions about how to prioritize the designs based on calculation and when to trust the results and when to not trust the results. And again, all those results were reported in this paper. You can read for more details. One important detail I want to emphasize here, that is titratable residues like ASP, gluon, and histidine may have multiple possible protein states. And the preferred protein state may differ between the protein-protein binding interface versus the unbound form. And the above results that is presented were after rigorously accounting for the multiple possible protein states according to the PK calculation of those residues in different protein environments. And accurate calculating protein PK is not only important for protein-protein binding potency prediction, it's also important for achieving pH-dependent binding. Antibodies with pH-dependent binding profile might be recycled through interaction with FcRn or might accumulate in cancer cells due to different pH environments between cancer and normal cells, providing a unique therapeutic advantage. And the key for all those applications is the accurate calculated protein PK in different environments. Therefore, we spend a lot of effort developed the accurate, reliable, constant pH method for protein PK calculation. As you hear from Wei's poster and talk on Monday, the accuracy for constant pH PK calculation for those type of residues is looking very good. And recently, we have also extended the method to calculate PK for other residues like lysine, cysteine. I want to emphasize cysteine here because cysteine is a classical target for covalent warhead. And the PK of cysteine is believed to be correlated with the reactivity or the propensity for the residues to form covalent bond. And here, with the systematic improvement in the force field, you can see that the accuracy for cysteine PK is also looking very promising. So I talked about the pH-dependent bonding between antigen and antigen antibody, providing unique therapeutic advantage. And we have also been applying constant pH and protein FTP to model the pH-dependent protein-protein binding. So here's one example. So based on the different pH environment between cancer and normal cells, scientists proposed to design an antibody targeting the HER2 mutant in the cancer cell. Without computational guidance, they randomly introduced a number of histidine in the binding interface. And only a few of them has the desired effect of improved binding at lower pH. Using constant pH and protein FTP, we modeled the effect of mutations on the protein-protein binding affinity at both neutral and lower pH values. And then consequently, we can also model the effect of pH environment on the protein-protein binding affinity. As you can see that both the effect mutations and pH sensing profiles are actually modeled. Here's the similar example. I'm not going to go to details. I want to show you a prospective application of constant pH and protein FEP for the design of pH-dependent disassembling of protein cage for drug payload delivery. So this is a collaboration. The collaborator identified a protein cage that has a cavity inside that can embed small molecule drug payload. The idea is to introduce mutations on those protein-protein interface so that the protein cage remains stable at neutral pH, but then become unstable at lower pH value. So they can disassemble the protein cage to relieve the drug payload, the cancer environment. You can see this is a very, very complex design problem. So we first apply MMGSA residue scanning to identify the hotspot on the protein-protein interface, then use constant pH and protein FP to model the effect of mutations on the protein-protein binding affinity at different pH values. And based on the calculations, we suggested 20 designs for experimental testing, including some control designs just to prove how the protein cage disassembling is affected by the calculated protein-protein binding affinity DDG. So here's the results. So first of all, we do see that there is a correlation between the experimentally observed protein cage disassembling proportion with the calculated protein-protein binding affinity DDG. And many of the designs indeed show a pH-dependent disassembling profile, like the example shown here. And so far, 7 out of 20 designs were confirmed by experiment to display those kind of pH-dependent disassembling profile, including 2 designs that has the best profile in the field. The collaborator is super excited by the novel designs from these calculations. So the protein FP method I mentioned earlier can only model 1 mutation per calculation. So in many biologic design projects, we may want to explore a large number of mutations from large number of residues. So we may not afford to run protein FP for all the combinations of mutations. So to address this limitation, recently we also implemented a new method called FP+ residue scanning for lambda dynamics enhanced protein FP. This is very similar to the multi-site lambda dynamics for small molecules that introduced by the speaker yesterday. I'm not going to spend too much time on the methods here. But in summary, this method provides a significant speed up as compared to protein FP without compromising accuracy. For example, we have validated this method on a large number of systems, including protein-protein binding, protein thermal stability calculation, and it achieved a similar level of accuracy as protein FP with only a very small fraction of time. And I want to wrap up by showing you one example about how those different complete methods can speed up the antibody drug discovery project in practice. So here, in this collaboration, the collaborator wanted to introduce mutations to improve the binding potency and selectivity with antigen. So they explored a large number of mutations, including all the 39 residues in the binding interface. Each one of them were mutated to all the other 18 residues, except for sensitivity to avoid reactivity. And the whole exercise only identified 33 mutants with improved binding. But the whole process took a very, very long time, longer than half a year, and very, very costly. So if we use competition approach, we can first run the FP residue scanning to score all the mutants, and then take the top few hundred for protein FP, and then the top like 96 for experimental testing. This whole process only takes about 2 weeks to finish, and then the cost can be reduced to be about 20%, but still recover almost all the positive mutations. So I know I'm running out of time, so I'm going to skip the summary for this section, and also want to highlight that we have also developed methods that can accurately and reliably predict small molecule crystal structures and solubility calculations to help small molecule drug formulation. You can read more details of the methods and validation in those papers. And I want to stop here and take any questions.
We're running a little behind this morning, so I'm going to stick to one question.
So if I understand you correctly, you're talking about the GPCR functional response modeling. How does the calculation results compare with experiment for the binding potency, right? Yeah. So yeah, I only show the data for the delta G, that is the calculated binding fitness between the active versus inactive state, then to predict the function response. But we also in the paper also do detailed comparison between the predicted DG for all the agonists, for example, as compared experimental results. They also have a very high correlation. And one caveat here is that experimental binding potency measurement for GPCR is very tricky. Most often they only report the indirect measurement, the percentage of the function response in most cases. So it's not quite kind of a apples to apples comparison with the calculated DG.
Let's thank Dr. Wang one more